My dear colleagues, welcome to the 4th International Erasmus Conference on Biological and Chemical Sciences. My name is Burcu, and I want to present you our study, which is called Alpha-lipoic acid prevents valproic acid-induced oxidative damage in lens tissues of rats. And we are participating to the Congress from Istanbul University, Cerrahpaşa. I want to give some brief titles about our presentation. We will begin with the introduction part, valproic acid, alpha-lipoic acid, and lens. And we will continue with the materials and method parts, and we will share our results, and we will conclude our presentation. Valproic acid is an effective drug which is preferred for the primary medication of epilepsy both in adult and younger individuals. And valproic acid-induced toxicities have been related to its free radical producing effect. A report indicates that anti-epileptic drugs and valproic acid causes elevation of free radical, leading to excessive oxidative stress levels. And the third part, we can see the structure of valproic acid. The negative effects include toxicities on liver, lungs, kidney, brain, and gastrointestinal system. Alpha-lipoic acid is a powerful antioxidant molecule and it is found in meat, spinach, and cabbage. Alpha-lipoic acid is an essential cofactor for mitochondrial enzymes. And this metabolic antioxidant has a special effect as scavenging free radicals both in hydrophilic and lipophilic environments. This molecule can catch free radicals by its reduced form dehydrolipoic acid and prevent oxidative damage. And at the third part, we can see the structure of alpha-lipoic acid. Lens tissue is a very close system and has unfortunately limited capability for repairing or regenerating itself. Altered glucose balance, related oxidative stress, affected polyol pathways are important signals for the construction of ocular damage. In this study, we investigate the protective effects of alpha-lipoic acid against valproic acid-induced lens damage in rats. And here is our materials and methods part. We have 4 groups. The first group is control group, and the second group is alpha-lipoic acid group, and the third group is valproic acid group, and the fourth group is combination groups. We administered alpha-lipoic acid 50 mg per kilogram for 15 days, and we administered valproic acid 0.5 gram per kilogram for 15 days to rats by intraperitoneally. And the fourth group received the chemicals at the same dose and duration. And on day 16, rats were sacrificed and lens samples were taken. They were prepared as homogeneous rats and then centrifuged. And we determined reduced glutathione levels, lipid peroxidation levels, protein carbonyl levels, glutathione transferase and superoxide dismutase activities, glutathione peroxidase and reductase activities, aldose reductase and sorbitol dehydrogenase activities, and we also determined the total protein levels of all tissues. Our results were evaluated using an unpaired t-test and analysis of variance using the NCSS computer package. Here is our results. At the first table, we see the lens reduced glutathione, lipid peroxidation, and protein carbonyl levels of all groups. Administration of valproic acid decreased glutathione levels, increased lipid peroxidation, and protein carbonyl groups. Administration of alpha-lipoic acid to valproic acid group reversed these levels statistically significant. And the second table, we see the lens glutathione transferase and superoxide dismutase activities of all groups. Administration of valproic acid decrease glutathione transferase activities and increase superoxide dismutase activities in control groups. After alpha-lipoic acid administration, the activities were reversed in valproic acid group. And the third table, we see glutathione peroxidase and reductase activities of all groups. Administration of valproic acid increased both of the activities in control groups, and administration of alpha lipoic acid decreased the activities. And the last group is aldose reductase and sorptor the heterogeneous activities of all groups. Administration of valproic acid increased both of the activities, and alpha lipoic acid administration reversed these levels in valproic acid group. And we can resume the results at this figure. Before alpha lipoic acid administration, the levels activities reduced in valproic acid group is seen here, and the levels and the activities increase in valproic acid group can be seen here. And after alpha lipoic acid administration, the levels and activities were reversed. As in conclusion, according to our results, we can say that it was concluded that alpha lipoic acid had a protective effect against valproic acid-induced lens damage. And this study was supported by the Scientific Research Projects Unit of Istanbul University, Cerrahpaşa. And here is our references which we use in the presentation. And we do thank you so much for your attention.